Well, thank you, Lena, for your kind words. I will try to be enthusiastic at this early hour of Wednesday morning. I think it's fitting that I start by paying homage to August Krogh. I don't know what he would think about my presentation today. He would probably regard it as a gross show of incompetence. But I know that he knew about the pancreatic islets because he met with Bunting and Best when he was traveling in North America after getting the Nobel Prize, and he was actually the person who imported, if I may say that, insulin to Europe and Denmark. His own work, of course, was devoted to the regulation of capillary blood flow in various tissues, I think mainly in the lung. And so he might have been intrigued to know a little bit about the regulation of blood flow in the pancreatic islets, which I showed on. This is the blood flow in the pancreatic islets. And although the pancreatic islets only comprise 1% of the pancreatic mass, they receive 10% of the pancreatic blood flow. So there is possibly there would have perhaps been a suitable preparation for August Krogh to work on the pancreatic islets. However, this is not the topic of my talk today, which is rather devoted to the role of ATP-regulated potassium channels in the pancreatic alpha and beta cells, and the opposite and paradoxical roles they play in these two cell types. So why should we bother about insulin and glucagon secretion, or the alpha cells and beta cells? Well, the insulin and glucagon, of course, the two major glucoregulatory hormones of the body. The insulin is secreted by the beta cells, which are red in this slide, and glucagon is secreted by the blue alpha cells. And in addition, there are somatostatin-secreting delta cells within the pancreatic islets. And I won't consider the delta cells today, but I will focus on the alpha cells and the beta cells. And the role of glucagon and insulin is to reciprocally regulate the plasma glucose concentration in the animal or in the individual. So, for example, when we start from normal glycemia, and if we exercise or if we fast, there will be hypoglycemia. So the plasma glucose concentration will fall below the normal range. And to combat that, glucagon secretion is increased and insulin secretion is switched off, and this leads to glucose production by mobilization of glycogen in the liver, and thus restoring normal glycemia. Conversely, when we eat, we produce hyperglycemia, and this will stimulate insulin secretion and switch off glucagon secretion, and that will then promote glucagon synthesis, the deposition of glucose in the liver, and thereby restoring normal glycemia. So insulin and glucagon, they play key roles in the regulation of the plasma glucose concentration. But and of course, for that reason alone, it's important that we understand the physiology of the release of these hormones. But it turns out that it's also very important for the understanding of type 2 diabetes, a disease that now affects 5 to 10% of the population in westernized society. And it's well recognized that diabetes involves insufficient insulin secretion for the level of plasma glucose levels. But it also, which is perhaps less known, involves abnormal glucagon secretion. And the defects of glucagon secretion are of two kinds. There's too much glucagon being secreted at high glucose when it is not needed, and too little glucagon secretion at low glucose when it's acutely needed. And in fact, impaired glucagon secretion in response to hypoglycemia is one of the major factors for mortality, or the increased mortality in insulin-treated diabetic patients. So it's very important for these reasons that we know more about the regulation of glucagon secretion. So what is known about the regulation of glucagon secretion? One of the most popular concepts in this field is that the alpha cell is not sensing the changes in the plasma glucose concentration itself. It's rather being under paracrine control by factors released from the beta cells and the delta cells. And as you can see here, this sort of arrangement of this regulation is facilitated by the close proximity of the alpha cells to both the beta cells and the delta cells. But if this is going to work, then it requires that that glucagon secretion is regulated over the same range as that regulating insulin secretion. And how well is this criterion fulfilled by the glucagon secretion? So to address this, we measured insulin secretion, and there's a black trace here, and glucagon secretion from groups of freshly isolated pancreatic islets at various glucose concentrations. And what we observed was that glucagon secretion is maximally inhibited by glucose concentrations at which there is little or no stimulation of insulin secretion. And we can make a similar argument for somatostatin. So this, I think, suggests that the paracrine regulation, I mean, I don't debate that there exists a paracrine regulation of glucagon secretion, but not in the terms of the control of the response to hypoglycemia. So the lack of an overlap between insulin and glucagon secretion and somatostatin secretion, to me at least, suggests that there must be some sort of intrinsic regulation of glucagon secretion, that the alpha cell itself has the capacity of sensing changes in the plasma glucose concentration and adjust the release of glucagon secretion accordingly. So what is known about this control, or this cellular control, intrinsic regulation of glucagon secretion? Well, we know that calcium is a very important regulator of secretion in many cell types, and that this is also true for the pancreatic islets. So here we have measured insulin, calcium using confocal microscopy in alpha cells and beta cells when we go from 1 to 6 millimolar glucose in intact pancreatic. So this is exactly the same preparation we used for the hormone measurements I showed you in the previous slide. And as you can see here, when we stimulate, we increase the glucose concentration to 6 millimolar, the beta cells respond, at least a fraction of the beta cells respond at this low glucose concentration with oscillations in calcium. And they are, of course, low to begin with, consistent with the initiation of insulin secretion by an elevation of glucose in the islets. But if you look at the alpha cells, we can see that they are spontaneously active already at 1 millimolar glucose, which is, of course, what we expect, given that glucagon secretion is high under these conditions. But the surprise is that when we look at the responses to 6 millimolar glucose, when we get a maximum inhibition of glucagon secretion, then hardly anything happens. If anything, these oscillations tend to be slightly bigger. So how can we explain this? I mean, what I would try to argue is that these calcium oscillations, they represent the wrong type of calcium, as I will try to show you later. So based on this sort of work and the work of many laboratories over the last 30 years, a consensus model for the glucose regulation of the pancreatic beta cells has been proposed. And this postulates that glucose is taken up into the beta cells, it's metabolized, there's an increase in the ATP-ADP ratio, and this in turn leads to the closure of ATP-regulated potassium channels. These channels, they normally maintain a negative membrane potential in the beta cells, so when they close, there will be a slight depolarization, which is sensed by the voltage-gated calcium channels. They open, calcium goes into the cell, and that this increase in calcium then triggers insulin secretion, and the insulin then acts, and then when insulin has acted and glucose has been removed from the plasma, this whole sequence of events is reversed and insulin secretion ceases. So this model, it is now been called the consensus model of glucose sensing in the beta cell. Now what about the alpha cell? Well, in the alpha cell, there is much less is known. We know many of the players, we know that there is glucose transporters, we know that there are calcium channels, we know that glucagon is released by vesicular excitosis. But we don't know anything about the link between this an elevation in plasma glucose and the suppression of glucose secretion. There are almost as many hypotheses explaining or trying to explain how glucagon secretion is regulated as there are investigators working on this problem. So we might call this the lack of consensus model of glucagon secretion. Now, one thing I should point out here is that the alpha cells, like beta cells, they have the ATP-regulated potassium channels, exactly the same type of potassium channels as those that mediate the glucose-induced depolarization in beta cells and that mediate or underlie the stimulation of insulin secretion in response to high glucose. So what are these channels doing in the alpha cells? I mean, how could possibly glucose-induced closure of these channels in the alpha cells mediate inhibition of glucagon secretion whilst mediating stimulation of insulin secretion in the beta cells? So to address this, we perform insulin or glucagon secretion measurements using a perfused pancreas preparation. So we started the experiment at 6 millimolar glucose, and then we lowered the glucose concentration to 1 millimolar. And as expected, this leads to a very marked stimulation of glucagon secretion in the perfused pancreas. And then we apply tolbutamide, which is a drug, a sulfonylurea drug, which has been used in the treatment of type 2 diabetes for now almost 60 years. And as you can see over here, this application of this drug, which is also a specific blocker of the ATP-regulated potassium channels, it leads to very dramatic and rapid and maintained inhibition of glucagon secretion. And it's interesting to note that glucagon secretion in response to tolbutamide is as strongly suppressed as it is in response to 6 millimolar glucose. So based on this, it seems that a stimulation of glucagon secretion in response to hypoglycemia is mediated by activation of the KATp-channels, and that inhibition of the KATp-channels, whether by glucose or by tolbutamide, leads to inhibition of glucagon secretion, which is a slight paradox. This is completely the opposite to what we see in beta cells, where inhibition of the channels leads to stimulation of secretion, not the other way around, as appears to be the case in alpha cells. So we applied electrophysiology to trying to understand how a closure of potassium channels in the alpha cells could mediate inhibition of glucagon secretion, and we applied patch clamp recordings to alpha cells in intact, isolated pancreatic islets, and then we recorded electrical activity, and as expected, from the fact that the alpha cells secrete glucagon at low glucose, these cells are electrically active at 1 millimolar glucose. And if you look carefully, you can see that the electrical activity in the alpha cells is quite different from that we observe in beta cells. And it has large overshooting action potentials. And we have carefully characterized the membrane currents underlying this electrical activity, and we have found that it results from the activation of 4 to 5 different types of ion channels, but suffice to emphasize the involvement of voltage-gated TTX-sensitive sodium channels here and high voltage-gated PQ-type calcium channels, and the PQ-type calcium channels are open at the peak of the action potentials, and the significance of that, I hope, will become evident later. There are also other types of high voltage-gated calcium channels, like the L-type calcium channels, but they don't seem to be that tightly linked to glucagon secretion. So now we applied membrane potential recordings to alpha cells in intact islets, and we started in 1 millimolar glucose, and then we tested the effects of tolbutamide. What are the effects of tolbutamide on alpha cell electrical activity? And I remind you that this concentration of tolbutamide, this will produce very strong inhibition of glucagon secretion. And much to our surprise when we did these experiments, there was absolutely no effect. There was no inhibition of electrical activity in response to tolbutamide. But we could observe that there was the alpha cells depolarized in response to the to tolbutamide, and this effect was reversible. We also observed that there was an increase in the action potential frequency. And then finally, as shown over here, there's also a decrease in the peak amplitude of the action potentials in the presence of tolbutamide. This is action potential 2, which is taken from this plot here. And down here we have the statistics, and you can see that tolbutamide on average depolarizes the cell by 9 millivolts. This is this change from here to here, and it reduces the spike height by about 16 millivolts, and it increases the frequency of the spikes more than twofold. So at least this shows the effects of tolbutamide. It shows that the KATp-channels are indeed active in intact alpha cells in alpha cells in intact pancreatic islet, something which has been questioned before. But I mean, I think this provides fairly conclusive evidence that the KATp-channels are there and they are capable of influencing the membrane potential of the alpha cells. Now, we repeated the same experiments with glucose, and now we used 6 millimolar glucose, and as you might recall, this is a maximally inhibitory glucose concentration, and the responses to glucose are very similar to those that we obtain in response with tolbutamide. We see a 9 millivolt depolarization in response to the glucose of the interspike membrane potential. We see an increase in spike frequency about twofold, and we see a reduction of the spike height by about 10 millivolts. So very similar effects are produced by glucose and tolbutamide. And collectively, these observations suggest that glucose and tolbutamide, they influence electrical activity by closing the KATp channels that are active in the alpha cells at low glucose. So, of course, this is only an indirect measurement, so the most direct way of assessing this would be to monitor KATp channel activity directly, and we have done this. We have done so by using perforated patch measurements in alpha cells within intact pancreatic islets, and in this approach, the cells are metabolically intact, so we can measure any glucose effects of glucose metabolism on KATp channel activity. And we monitor KATp channel activity at a host cell level, applying small voltage pulses around the resting potential of the cell, which we, in this protocol, set to minus 70 millivolts. And then we measured potassium or KATp channel activity in these cells, and these are the results. So when we apply, when we start in 1 millimolar glucose, we measure a conductance which is about or less than 300 picosiemens. When we apply glucose, we have observed a small decrease of 20-25% decrease in conductance from 280 to 230 picosiemens in response to 6 millimolar glucose. We observe exactly the same reduction when we apply tolbutamide, which I remind you is a specific blocker of the KATp channels. And what we have plotted here, I should point out, is we measured the conductance, which is obtained by dividing the current responses by the voltage pulses that we applied in the, as illustrated in the previous slide. And a decrease in conductance that corresponds to an inhibition of KATp channel activity. So this decrease here corresponds to inhibition of KATp channels. I should also point out that when we add glucose in the presence of tolbutamide, there is no further inhibitory effect. So that really shows that glucose acts on the ATP-regulated potassium channels. Now, what struck us was that these changes are extremely small. I mean, they're limited to 50 picosiemens. And I mean, just to illustrate or explain how small these changes are, 50 picosiemens, that's equivalent of about 4 active KATp channels in the entire alpha cell. So these are very small changes in channel activity. And this is also underscored when we compare the responses in alpha cells to the responses that we can obtain in beta cells under otherwise identical experimental conditions. If we do this sort of experiments in beta cells, we would see a reduction of about 5,000 picosiemens. So these changes we observe in alpha cells are only about 1% of those we observe in beta cells. And I should point out or explain that this is not because the alpha cells contain fewer KATp channels. If we establish a host cell configuration and wash out intracellular ATP to maximally activate the KATp channels, the total KATp channel activity that we can measure in the alpha cells is, if anything, much larger than that in beta cells. So that suggests that for reasons that we don't understand yet, the KATp channels in alpha cells are under very strong tonic inhibition. Okay, so now to measure or to establish the relationship between KATp channel activity and glucagon secretion over a wider range of KATp channel activities, we performed the experiment where we increased KATp channel activity pharmacologically using a compound called diazoxide, which we showed many, many years ago is a selective activator of KATp channels. So the way the experiments was done was that we started experiments in 6 millimolar glucose to maximally inhibit KATp channel activity and glucagon secretion. We measure the potassium channel activity expressed as membrane conductance and glucagon secretion from the same islets. And this is what we get at 6 millimolar glucose in the absence of any diazoxide. We then increase KATp-channel activity experimentally by applying progressively increasing higher concentrations of diazoxide, 1, 3, and 10, and 100 micromolar. And what you can see is that 1 and 3 micromolar, although they produce a very small increase in KATp-channel activity from 2 to 250 to 300 picosiemens, they stimulate glucagon secretion. But then when we go to higher concentrations of diazoxide, like 10 micromolar, when we have 500 or 600 picosiemens of conductance, or when we have 100 micromolar, you get a secondary inhibition of glucagon secretion. So there is a bird-shaped relationship between KATp-channel activity and glucagon secretion in alpha cells. And we can observe here that 3 micromolar diazoxide is roughly equivalent to 1 millimolar glucose in terms of its effects on KATp-channel activity and glucagon secretion. And you can also observe that the changes of KATp-channel activity in the physiological range or in the physiological conditions are extremely small. I mean, it goes from about 300 picosiemens maximally down to 200 picosiemens. This is the range where KATp-channel activity is regulated physiologically and that controls glucagon secretion in vivo, we would think. Now, and what can also be observed from this relationship is that any deviation, whether you go to the left or to the right, from this optimum around 300 picosiemens will be associated with inhibition of glucagon secretion. And I think that I will come back to that later because I think that has important pathophysiological implications. Now, there's an interesting thing here is if you compare the responses to 6 millimolar glucose in the absence of diazoxide and glucagon secretion at 6 millimolar glucose in the presence of a high concentration of diazoxide. They are both, glucose secretion is equally suppressed under both experimental conditions. And we then try to correlate this with the changes in membrane potential. And this is quite interesting because this is what we observe. So here we go from 1 millimolar glucose to 6 millimolar glucose. We get the inhibition or the depolarization of the membrane potential. You see the reduction of spike height. You see the stimulation of action potential firing. And then we apply but you remember that this is now where we get maximum inhibition of glucagon secretion. We then apply a very low concentration of diazoxide, which increases KATp-channel activity by 30 picosiemens, equivalent to 2 KATp-channels. And that reverses the effect of glucose. And then, as you observed in the previous slide, this stimulates glucagon secretion. And finally, we apply 100 micromolar diazoxide, which massively increases the conductance. And that, of course, then leads to hyperpolarization and the cessation of electrical activity. Now, the interesting thing is that if we compare glucagon secretion under these conditions and under these conditions, glucagon secretion is suppressed, paradoxically suppressed to the same extent. And whilst it's very easy to understand why glucagon secretion is low under these conditions, simply because there is no action potential firing, it appears as if these action potentials are useless when it comes to stimulating or initiating glucagon secretion. So why is that? And why don't these action potentials, which are fired at a high rate but at a lower amplitude, why do they fail to produce glucagon secretion? And this is a rather complicated story, and I don't have the time to discuss this in as much detail as I would like. But suffice to say here today that this reduction in spike height that we observe in response to glucose, it results from voltage-dependent inactivation of the sodium channels, which are important for the upstroke of the alpha cell action potential. And for that reason, and because the action potentials now stop at a more negative membrane potential than before, then there is less activation of the PQ-type calcium channels. And the PQ-type calcium channels are the ones that mediate the calcium that is necessary to initiate glucagon secretion. And just to illustrate how important the PQ-type calcium channels, we have done the experiments to the right where we have tested the effects of glucose in the absence and presence of agatoxin, which is a highly selective blocker of the PQ-type calcium channels. And these are the control conditions where we get nice inhibition of glucagon secretion with 6 millimolar glucose. If we apply agatoxin, you can see that agatoxin on its own is as strongly inhibitory as 6 millimolar glucose under control condition. And in the presence of agatoxin, when glucagon secretion is already suppressed, glucose has no further inhibitory effect. So that suggests that whatever the effects of glucose on electrical activity, they culminate in a reduced activation of the PQ-type calcium channel, and that in turn leads to suppression of glucagon secretion. We can illustrate this in a different way by measuring the ability of different voltage pulses to evoke excitosis, and to this end we used capacitance measurements of excitosis, which allow us to measure the changes in cell surface area as capacitance, which result when the cell surface area is increased by the insertion of new glucagon granules into the plasma membrane. And we can use this now to control the membrane potential and study in a very detailed fashion exactly how the ability of depolarization to evoke glucagon granule excitosis depends on the amplitude of the voltage pulses. And so here we have, this graph summarizes the responses of the voltage dependence of excitosis in the alpha cell. And you can see that as we go above minus 10 millivolts, excitosis increases very steeply in the alpha cells, and it peaks around plus 10 millivolts. Now the red arrow here indicates the decline in excitosis that you would expect from the reduction of action potential height in response to glucose. And I mentioned before that glucose produces a 10 millivolt reduction of the action potential height. And from this relationship, we can estimate that the reduction of action potential height will produce a 75% reduction of excitosis in the alpha cell. In the case of tolbutamide, there will be an even stronger effect because the effect of tolbutamide was, of course, even stronger on the amplitude of the action potentials. So this is now an attempt to summarize what I talked about so far, and this is just to explain how the beta cell responds to glucose. At low glucose, the beta cell has a high KATp-channel activity, and that keeps the membrane potential negative, and there's no action potential firing, and because of that, there is no initiation of insulin secretion. Now, when we apply 6 millimolar glucose, the KATp-channel, or high glucose concentrations for that matter, the KATp-channel is closed. There is membrane depolarization, electrical activity is initiated, and that leads to stimulation of insulin secretion. During electrical activity, there might be a slight decline in the height of the action potentials in the beta cells as well, but insulin secretion remains much higher even after the reduction of spike height has occurred than under basic condition, just because you start from this very low level. So, and this might then potentially explain the biphasic nature of insulin secretion. Now, in the alpha cells, the situation is rather different, because in the alpha cells, we have very low KATp-channel activity to begin with. It's low, but it's bigger or larger than zero. But this low KATp-channel activity, that allows the alpha cell to fire action potentials. And that accounts for a high rate, or relatively high rate, of glucagon secretion. Now, when we increase glucose, the KATp-channel, the remaining KATp-channels close, excuse me, and this leads to a transient stimulation of action potential firing, which might lead to a transient stimulation of glucagon secretion, similar to what some investigators have reported previously. But eventually, the sodium channels inactivate because of this membrane depolarization. There's a decrease in spike height, and this reduction in spike height, it leads to, and this occurs via the inactivation of the sodium channels, and this leads to a reduced activation of the PQ-type calcium channels. And for that reason, there is a reduction of glucagon secretion. I point out here that the L-type calcium channels, they continue to open during electrical activity, and they continue to mediate the calcium influx, and that might account for this dissociation between calcium and glucagon secretion, which I showed in one of my first slides. Now, it's interesting to do a little bit of mathematics on this. So if we know that the action potential frequency increased by 200% in response to glucose or tolbutamide in the alpha cells. But this increase in spike frequency is insufficient to compensate for the much greater inhibition of exocytosis that results from the reduction in spike height, which was a 75% reduction. So if we combine these two factors, we can estimate that glucose would produce a 50% inhibition of glucagon secretion, which is in fact very close to that we observed experimentally. Okay, so my clinical friends at Octum, they like to remind me that rodent diabetes is not a major clinical problem. And if there are mice in our diabetes clinics, it's a pest control problem. I've been reliably informed, rather than a medical problem or a diabetes care problem. So we thought that it would be very important then to translate these observations, which we have done in mouse islets, rodent islets, to the human situation. And in Oxford, we are in a good position to do this, because in Oxford we have in the same building where we work, we have the Clinical Islet Isolation and Transplantation Center, which is directed by Professor Paul Johnson. And since 2006, we have had the opportunity to study more than 160 pancreases, and we have measured glucagon secretion for many of these. And occasionally, we also get pancreases from type 2 diabetics. Now, now, as I pointed out before, or in one of my early slides, in diabetes, we have in addition to the defects of insulin secretion, we also have disturbances of glucagon secretion. And whereas in healthy people, ingestion of a high glucose meal, a large chocolate bar, for example, leads to inhibition of glucagon secretion, we observe the inverse response in type 2 diabetics. We get stimulation rather than inhibition of glucagon secretion. And this, of course, exacerbates the hyperglycemia that results from the lack of insulin in diabetic patients. And the interesting thing is that we observe exactly, if we do the experiments in islets taken from diabetic donors, we see exactly the same thing. So in non-diabetic donors, healthy donors, 6 millimolar glucose produces the same inhibition of glucagon secretion as we observe in rodent, about a 50% reduction. Now, if we take the responses that we have seen in 10 preparations of diabetic islets, we see that glucagon secretion at 1 millimolar glucose is, if anything, slightly reduced compared to what we see in the non-diabetics, and that glucose paradoxically stimulates glucagon secretion in diabetic islets, echoing the responses that people observe in clinical studies in vivo. And it's quite easy to understand. I mean, I explained this before, how hyperglycemia and hypoglycemia regulate glucagon secretion in non-diabetic islets. As I showed you in rodent islets, we postulate that there's a bell-shaped relationship between KATp-channel activity and glucagon secretion also in human islets, and we have data to support this. And again, when we increase the plasma glucose concentration, there is a reduction of KATp-channel activity, and we move down this descending limb of the relationship. And conversely, when we lower the glucose concentration to produce hypoglycemia, we reactivate KATp-channels, and that leads to stimulation of glucagon secretion. Now, it occurred to us that with this bell-shaped relationship, you could actually explain the inverted response to glucose in diabetic islets if you assume that for some reason there is an increase in the KATp-channel activity, perhaps because of a impaired glucose metabolism in the diabetic metabolism. So rather than sitting over here on the descending on the left limb of the relationship, you sit over here. And now, if you sit here and you increase the glucose concentration, well, of course, you inhibit KATp-channel activity, so you would move from here to here. But unlike what you observe in the non-diabetic islets, this reduction of KATp-channel activity would now lead to stimulation of glucagon secretion rather than inhibition. And conversely, when we lower the glucose concentration, we will get inhibition of glucagon secretion rather than what would be physiologically appropriate stimulation of glucose secretion. So just by moving the set point, if you like, from left to right, we can explain both the abnormalities that we see of glucagon secretion associated with type 2 diabetes. But of course, this is only hypothetical, and it's very important that we do the experiments to show that this actually occurs. And the way we did this was that we used diazoxide. You remember this compound that activates KATp-channels? We used a very low concentration of diazoxide, the same concentration of diazoxide that produces the same increase in KATp-channel activity as the inhibition normally produced by glucose. And again, if we do this in under controlled conditions, 6 millimolar glucose produces the normal inhibition of glucagon secretion. But if we repeat the experiments with the same islets in the presence of diazoxide, you can see that under these conditions, glucose secretion at 1 millimolar glucose is reduced compared to what we have in the untreated islets. And when we apply glucose, we get stimulation rather than inhibition of glucagon secretion. So that's quite interesting. Just by increasing the KATp-channel activity, we can recapitulate the defects of glucagon secretion seen in type 2 diabetes. What might be the reason for this? Well, we reason that perhaps there could be an impairment of glucose metabolism in diabetic islets. I mean, there is accumulating evidence now from work in other groups that this indeed is the case. So we tested this using a rather brutal approach. We used the mitochondrial blocker oligomycin. And as you can see, oligomycin, it has exactly the same effects as diazoxide or type 2 diabetes. It lowers glucagon secretion at 1 millimolar glucose, but it also results in this paradoxical stimulation rather than inhibition of glucagon secretion in response to glucose. And it's actually quite interesting, if you think about it, that the islets, they respond to glucose in the presence of the mitochondrial inhibitor. And somehow that suggests that non-mitochondrial metabolism might be very important for the control of glucagon secretion in alpha cells. Now, so this just shows that an impaired metabolism is important. But what is also exciting is that we can cure these metabolically compromised or poisoned, if you like, islets using tolbutamide. So these are exactly the same data as I showed you in the previous slide. We have the inverted glucose response. Now we treat the islets with a low concentration of tolbutamide. This is maybe only 1/10 of that we would normally use to stimulate insulin secretion. So it's a very low tolbutamide concentration. But what you can see here is that when we expose the poisoned islets to tolbutamide, we restore normal glucose competence in the poisoned islets. So there's nothing very wrong with the oligomycin-treated alpha cells. They remain capable of responding to glucose, and just by applying tolbutamide, we can restore the normal responses. Increase the glucose secretion at 1 millimolar glucose, and we have inhibition rather than stimulation of glucagon secretion in response to glucose. Now, of course, the important thing is, I mean, it's all very well to do these experiments in isolated islets, in poisoned islets, but of course the critical test of this would be to do the experiments in diabetic islets. And we were quite fortunate before Christmas last year because we got 4 diabetic preparations. So we could directly test this concept. So this is, and here we have plotted the percentage change of glucagon secretion when we increase the glucose from 1 to 6 millimolar. And this is the non-diabetic islets where we get an average of 40 to 50% inhibition of glucagon secretion. Now, if we take those diabetic preparations, then we get stimulation rather than inhibition of glucagon secretion. But what is very interesting is that if you take human type 2 diabetic islets and expose them to 6 millimolar glucose in the presence of tolbutamide, we normalize the glucagon response in these islets. So we actually increase glucagon secretion at 1 millimolar glucose, and we have the normal suppression by glucose. So how can we explain this? So this is what I think happens. So because of this, in the type 2 diabetes, we have a metabolic defect. So rather than sitting over here, we now sit over here. We expose the cells to glucose, and that leads to a reduction of KATp-channel activity. So we move from here to here. That results in stimulation of glucagon secretion that we observe. So that is what I just said. Now, if we apply tolbutamide first, then what happens is that we still start from over here. Tolbutamide, a low concentration, a concentration that blocks 50%, takes us from here to here. And tolbutamide itself would lead to stimulation of glucose secretion, which, of course, which is exactly what we observe. And then we apply glucose on top of tolbutamide, and then that will produce a further decrease. And under these conditions, you will move from here down to here, and you will get the inhibition of glucose secretion. So but the important thing is that you use a low concentration of tolbutamide, because if you use a very high concentration, you would go from all the way from here down to here, and you will have a complete inhibition of glucagon secretion. So if one wanted to use this information clinically, it is very important that much lower concentrations of tolbutamide were used than are traditionally used to cure the insulin secretion defects. So let's see. So this is what I've tried to say. I mean, I think that the alpha cells that secrete glucagon, they possess an intrinsic regulation, but that, of course, does not exclude a paracrine regulation under certain experimental conditions. I mean, maybe when glucose is much higher than the normal 6 millimolar, the paracrine regulation might become important. I think we also show that KATp-channels are active in intact alpha cells at low glucose, but the activity is very low, and that closure of the KATp-channels by glucose or tolbutamide, it leads to membrane depreciation and a reduction of action potential height. And this reduction in action potential height, it culminates in reduced activation of the PQ-type calcium channels, and that then accounts or explains the inhibition of glucagon secretion. And then finally, I showed you that the inverted glucose response that is a hallmark of type 2 diabetes can or might result from an increase in KATp-channel activity due to a metabolic disturbance, and that can be corrected by a low concentration of tolbutamide. And of course, it's always customary to have an acknowledgement slide which shows you the place where you work and the people who have done the work and the funding bodies. And this is, of course, a representative recording of Oxford, of beautiful buildings, and the sun is shining. But those of you who work in electrophysiology, you know, of course, that there is the representative recording is not always representative of the real experiments. I should show you what the reality looks like from our lab. This is what it looks like. And not so sunny, not so beautiful buildings. But having said all this, I hasten to add that I think that what I've told you today is actually an accurate representation of our experiments. And the experimental traces I've shown you, they are not selected. They are indeed representative, believe it or not. Thank you very much. And I should, of course, acknowledge the people that did the work. Xuan Shang is the person who's done all the electrophysiology, and this has been a tour de force. He's worked 7 years on this. I think he deserves lots of acknowledgement for this. And Reshma, she has done almost all the secretion measurements. And the other people here that have contributed in various ways to the project. And I should also acknowledge Paul Johnson, of course, for giving us the opportunity to work on human pancreatic islets. Thank you very much.